I want to welcome you. My name's Josh Miller. If you don't know, I'm the chair of the Department of Nutritional Sciences here in the School of Environmental and Biological Sciences. And today I'm welcoming you to one of the highlights of the academic calendar for our program, the Hans Fischer Lectureship. First of all, very excited. As usual, Hans and his wife Ruth are here, and that makes it extra special. I'm going to move right along. The first thing I'm going to do is introduce Executive Dean Goodman of the school to have some opening remarks.
Thanks, Josh, and good morning, everyone. Delighted to be with you on this annual auspicious occasion of the Hans Fischer Lectureship. So I'm very conscious of the fact that there are many new faces in the audience, perhaps. Many of you not here when Hans was in full sway at Rutgers and when he retired. So I thought my opening today would be useful if I very briefly, because it's an incredibly long biography, gave you a quick overview of Hans's life. So the lectureship, of course, was created to honor Hans and his many contributions to Rutgers, and as the founder of the Department of Nutritional Sciences here at Rutgers University in New Brunswick. Hans is a trailblazer whose accomplishments included many important discoveries about the role of diet and the mechanisms of alcohol, neurochemical changes that occur during addiction, and how those changes might be managed. He was also a pioneer in finding ways to control cholesterol and heart disease and in linking tryptophan and serotonin with behavior, a discovery that has led to very important treatments for both schizophrenia and alcoholism. So Hans was a young boy in Germany during the perilous times that led up to the Second World War. And he was shaped by those experiences in those times as only a child can be. On Kristallnacht in 1938, Hans's father was rounded up and taken away from his family. His father was a distinguished World War I veteran, winner of the Iron Cross medal, and was a couple of months later released from the prison camp and ordered to leave Germany within 2 weeks. That was a tall order for the family, and so Hans's father left on his own, alone, with a visa to Panama, and ended up in Cuba, where he awaited the arrival of his family following him. But that reunion, that first reunion that was planned, never happened, because the family, who were on a ship coming across the Atlantic, the ship's passengers were disallowed from entering Cuba. And finally, after a long saga, the ship returned to Europe, to Belgium, and the families were dispersed to France and England and Holland. Hans and his mother and sister were settled in France, but the family even then couldn't stay together because children of Hans's age were sent off to a children's camp. In this case, I believe it was in a suburb of Paris. He spent several months there and was finally reunited with his family in Laval, a small town in central France. And by the end of December of the following year, 1939, we're at now, they left France to Southampton in England and awaited on a ship that joined a huge convoy to cross the Atlantic, which by then was very perilous, to Ellis Island. But after being processed at Ellis Island, were not allowed to stay. They went aboard the next ship to Havana, where the reunion with Hans's dad finally happened. He spent a little over a year in Havana, leaving in February 1941 to come back to the States, to New York. So at this time, Hans was 12 years old. So a lot happened in those first 12 years of his life. Unable to find work in the city, Hans's father decided to take up poultry farming. And he moved the family to Vineland, New Jersey in September of that year. Time proceeds quickly now in this narrative. Now it's 1946. Hans enrolls at Rutgers as an undergraduate, where he met a couple of professors who he's written in the autobiographical article from which this photo is taken, and from which my comments are abstracted. A couple of professors who, quote, made a greater than average impression on the young man. And he became a nutritional biochemist, joining the faculty in 1954. So Hans spent more than 52 years at Rutgers, if you count the years up until his retirement. Founded the Department of Nutritional Sciences in 1966. Continued on for another 22 years. He served for a time as Associate Provost for the Life Sciences at Rutgers. And now he's here with his wife, Ruth, joining us again for the Hans Fischer Lectureship. Hans and Ruth, it's thrilling to have you here and to see you so well. And to you all, I give you Hans Fischer.
I will continue the biographical sketch. This time it's the biographical sketch of the department. Rutgers, from fairly early on, started a program of evaluating all departments, centers, institutes. And as a matter of fact, I chaired that committee for a very long time. And very outstanding academics from throughout the United States were brought here to make an academic evaluation of the teaching and research programs. And in 1990, 28 years ago, this department was reviewed under the chairmanship of Dr. John Suttie from the University of Wisconsin, member of the National Academy of Sciences. At that time, he was chairman of the Madison, Wisconsin Department of Nutrition and also a member of the biochemistry department there. He was an expert on vitamin K metabolism. Some of you may know that. And the department got very good marks from that review. And one of the recommendations was the establishment of this lectureship. The committee stated that the department and the college and the university would benefit from a lectureship that would bring outstanding researchers to the university on a regular basis, and at the same time to honor me for my contributions to the university and the department. So that's how it got started. As you heard, it got started in 1966, and in retrospect, I think it's comical to tell you that when the department was started in 1966, the dean was furious because there had been a total of 34 or 35 departments here at what was then Cook College, and the dean had just whittled them down to 17. because there were so many tiny little units, and he wanted to combine some of these units and make them more efficient. And here comes the university president, Dr. Mason Gross, who gives him the order to start a new department with one single faculty member. I was elected, by the way. There was a nutrition council at Rutgers at the time that included about 14, 15 people with an interest in nutrition from many different departments throughout the university, not just from this college. And that committee voted to have me chair the department. And so, as I said, the dean was very, very unhappy. He didn't talk to me for 1 year because he thought that I had gone to the president and had talked him into doing this. But I swear I had never talked to the president. I didn't know the president personally at all. In any case, that's how the department got started. The chair of the Department of Animal Science, of which I had been a member, was extremely accommodating, and he provided secretarial help and some additional funds for the nitty-gritties of running a department. And the dean overcame his anger, and the following year, 2 other people were permitted to join the department, and things became more normal, and the dean left. That really made it much easier. Anyway, I am so very happy that we have Dr. Nestler talking today. And just to show you what a small world it is, one of my sons asked me the other day who the speaker was. I said, it's Dr. Eric Nestler. He said, oh, I think I know who he is. We have collaborated with him on a recent research project. So on that note, thank you very much for coming, and I know we will greatly enjoy and learn from Dr. Nestler.
Okay, it's my pleasure now to introduce to you the chair of our seminar committee, Dr. Tracy Anthony. And as she's making her way up, I also want to quickly thank Laura Amador, who puts these events together, and as well as the seminar committee for getting such a good choice.
Yeah, I just want to point out in the seminar committee, Sue Shapsis, Joe Dixon, and Malcolm Watford. And I know Joe Dixon in particular did a lot of the heavy loading and idea to bring Dr. Nestler here. And so I just want to give thanks to that committee, in addition to Laura, who's great. She's the best ever. She did a lot of work for this. So with that, it's really my privilege to introduce to you Dr. Eric J. Nestler. Dr. Nestler is the Nash Family Professor of Neuroscience at the Icahn School of Medicine at Mount Sinai in New York, where he serves as Dean for Academic and Scientific Affairs and Director of the Friedman Brain Institute. He received all of his degrees, his bachelor's, his doctoral, and his medical degree, plus psychiatric residence training from Yale University. And then he served on the Yale faculty, where he ultimately was the Elizabeth Mears and House Jameson Professor of Psychiatry, Pharmacology, and Neurobiology, and Director of the Division of Molecular Psychiatry. He then moved to Dallas, where he was the another named professorship, Lou and Ellen McGinley Distinguished Professor and Chair of the Department of Psychiatry at the University of Texas Southwestern Medical Center. And then he finally came to New York. That was about 10 years ago. So Dr. Nestler is a member of the National Academy of Medicine. He is a fellow of the American Academy of Arts and Sciences. He is a founder and scientific advisory board chair for psychogenetics and a member of the board of directors of Berg Pharma. He is the author of over 500 publications and 5 books. Now, the goal of Dr. Nestler's research is to better understand the molecular basis of drug addiction and depression. His research uses animal models of these disorders to identify the ways in which drugs of abuse or stress change the brain to lead to addiction or depression-like syndromes, and to use this information to develop improved treatments of these disorders. We are honored as a department and privileged to have him here today as our Hans Fischer Lectureship speaker and have him share with us his thoughts on transcriptional and epigenetic mechanisms of depression. Dr. Nestler.
Thank you so much. It really is a great pleasure to be here. And as Hans mentioned, a particular treat for me to meet Hans and Ruth because I have been collaborating with Hans and Ruth's older son for several years, David, who's chief of dermatology at Massachusetts General Hospital in Boston. And also because when I was a graduate student and entering the field of neuropharmacology, I read Hans's papers on tryptophan and serotonin metabolism. So it really is a particular pleasure to be here and help celebrate this wonderful event. What I'd like to do today is to give you an update on some of the work that we've done in the area of depression, and I'll start by putting this in some clinical context. So it's impossible to overstate the impact that depression has on humanity. The World Health Organization has determined that depression is the #1 cause of disease, disability worldwide, and really an astounding statistic. On the other hand, we still know relatively little about what causes it. It's thought to be about 35% heritable, so it's not as heritable as other psychiatric syndromes like schizophrenia, bipolar disorder, or autism. For that reason, environmental factors are also thought to be very important. For example, chronic stress is implicated in some people, but not in others. And the way we view depression today is not as a single disease entity, but instead a heterogeneous collection of many diseases. It's a broad syndrome that really remains vaguely defined. We do have some good treatments for depression, antidepressant medications, psychotherapies, but large studies in the last decade have shown that only about half of all depressed patients respond fully to the range of treatments that are available today. Also, all of the medication treatments that are available today, including the newer medications like the SSRIs, all have the same mechanism of action as the first antidepressants that were discovered by serendipity 60 years ago. So there's an enormous unmet need to better treat depression. We've learned a lot over the last decade by use of brain imaging in identifying parts of the brain that are important in controlling mood under normal circumstances and perhaps represent the site of pathology in depression. But unlike syndromes like Parkinson's disease, where a specific type of nerve cell in a single brain area has been implicated, depression is thought to involve a broad circuitry within the brain. Some people refer to it as the limbic or emotional circuitry, involving broad regions of prefrontal cortex, a reward center that my lab has studied a lot, the nucleus accumbens, in the temporal lobe, the amygdala and hippocampus, among other regions such as hypothalamus and the site of the brain's monoaminergic neurotransmitters, for example, the ventral tegmental area, providing dopaminergic innervation of the limbic forebrain. So that's by way of very quick introduction to human depression. I'm a molecular biologist by analogy with all other fields of medicine. Most advances have occurred through the use of animal models, and that raises the notion of an animal model of depression. And I think that this difficulty in establishing an animal model of a psychiatric syndrome is at the core of one of the reasons why it's been harder to make progress in psychiatry than in most other medical specialties. Today in psychiatry, we still define syndromes solely on the basis of behavioral abnormalities. We still lack objective biological tests, blood tests, brain scans, genetic tests that inform a specific diagnosis or prognosis. And that means that we have to focus on these behavioral abnormalities because in the sense that's the only thing that we have to anchor the animal studies to the human syndromes. So I'll be mentioning a few animal models or chronic stress models to you today. I'll start with something that we call chronic social defeat stress. This was introduced into the lab by Olivier Berton, who's now at NIDA, and Nadia Tsankova, who's at Mount Sinai. And what they did over 10 years ago now was to take a genetically normal inbred C57 mouse, put it in the cage of a bigger, meaner mouse, a retired CD1 breeder. Fighting ensues instantaneously. We limit the physical encounter to just a few minutes to reduce physical injury, and then separate the animals with a screen for the rest of the day so that that test mouse is subjected to all the aggressive cues of the larger mouse. And then we repeat that every day, typically for 10 days. This is a high level of a combination of physical and emotional stress experienced by that test animal. And by the end of that 10-day period, we have defined a syndrome, behavioral syndrome exhibited by these animals. This would be equivalent to a mouse diagnostic statistical manual for social defeat. And we say that the animals exhibit anhedonia, or reduction in interest in pleasurable activities. I think we can measure that well in a rodent. Animals show decreased interest in drinking sugar water, eating high-fat food, having sex, running on an exercise wheel, and so on. We say the animals exhibit anxiety-like symptoms, but all measures of anxiety in rodents are really a derivative of exploratory behavior. These animals exhibit less exploratory behavior. They show hyperactivity of the hypothalamic pituitary adrenal axis, increased glucocorticoid secretion in response to certain circumstances. All mammals exhibit circadian rhythms and a variety of physiological functions. For example, temperature fluctuations throughout the day. These mice show disrupted rhythms. They are more prone to learn to self-administer and addict themselves to drugs of abuse. They exhibit an interesting metabolic syndrome where even though we interpret that they enjoy eating less, they eat more and induce obesity in themselves, even to the extent of developing central leptin and insulin resistance. And that may capture some of the comorbidity that's been described in subsets of people with depression with obesity, and profound social avoidance. Social defeat is an interesting paradigm to us for a few features. First of all, only about 2/3 of mice subjected to the course that I described in the previous slide show the full range of symptoms that I just spoke about. We refer to them as susceptible. The remaining third avoid most of the symptoms. They show equivalent decrements in exploratory behavior, but avoid all the other range of behavioral abnormalities. We call them more resilient. This makes social defeat somewhat unique among chronic stress models in enabling us to parse whether an adaptation to chronic stress is adaptive or maladaptive. Secondly, unlike most other chronic stress models in adult rodents, where the abnormalities are rather transient, the animals recover behavioral function relatively quickly at the end of stress, some of these abnormalities induced by 10 days of social defeat stress are permanent. And this range of permanent abnormalities can be reversed by the same range of medications that work to treat depression in humans. Anxiolytic drugs like benzodiazepines, for example, are without effect. Because of the stability, the ability of chronic stress to induce essentially lifelong behavioral abnormalities, we had proposed years ago that perhaps changes in gene expression are one important mechanism underlying those features. And that notion is depicted in this slide. We know that a mammalian organism has about 3 billion nucleotides in its DNA. If stretched out linearly, that would be about 2 meters of DNA. And somehow, 2 meters of DNA is compacted to an extraordinary degree to fit within a microscopic cell nucleus in every cell in the body. And we've learned a lot about that state of compaction over the last 10 or 15 years, a field referred to as epigenetics. So that's shown in cartoon fashion in this slide, where the DNA double helix is wrapped around octamers of histone proteins to form the unit of chromatin. Chromatin refers to just the material in the nucleus, called the nucleosome, which then get further organized in the most compacted state to what we recognize as a chromosome. And this is important because the state of compaction of DNA determines its ability to function. DNA that's in highly compacted regions of chromatin cannot provide biological activity, whereas DNA in more open regions of chromatin can provide biological activity, for example, encoding RNA and protein. And for that reason, we've been very interested in taking advantage of this knowledge to help us identify genes affected by stress, to understand mechanisms that mediate the ability of stress to produce changes in gene expression. By analogy with the cancer and developmental biology fields, where certain types of epigenetic changes, some types of changes in the compaction of chromatin are essentially permanent once they occur, we hypothesized years ago that maybe the same thing occurs with behavioral experience. Then in response to behavioral experience, some of the changes in chromatin structure are permanent, leading to lifelong changes in behavior. And the hope is that this would somehow inform our ability to better diagnose and treat depression. Now, I'm going to focus on this level of analysis, and this is the most complicated slide I'll show, providing a little more detail about what's known about the mechanisms underlying this compaction or loosening of chromatin within a cell nucleus. It depicts the two ends of the spectrum of chromatin, active open, where genes can encode RNA, or inactive condensed, where the DNA is too compacted to provide a biological function. In open areas of chromatin, relatively spare areas of DNA can bind transcription factors, proteins that bind to DNA in a DNA sequence-specific manner, recruit up to hundreds of other proteins into the vicinity of that binding site to induce transcription. The repression of transcription is equally complicated, where the condensation of chromatin is mediated by a similar level of a number of proteins called repressors that mediate that compaction. And one of the features that are involved is post-translational modifications of the histone proteins themselves, or DNA, for example, acetylation of histones, phosphorylation, methylation, or methylation of DNA itself, as just some examples of the mechanisms governing this opening and closing of DNA. So starting at about 10 years ago, when we moved to New York, back to New York, we decided to take a step back, and instead of focusing in on a given protein or gene of interest, to take a more open-ended view of what might be happening in our chronic stress models or in human depression, based on the notion that perhaps we don't know enough about the biology of stress or depression to always guess correctly what gene or protein to study. So that approach is depicted in this slide, showing the use of multiple platforms of next-generation sequencing data, focusing on changes in gene expression and associated changes in chromatin biology. So we would begin by RNA sequencing, RNA-seq, to identify sets of RNAs that are up or down regulated in a given experimental condition. If an RNA, for example, is induced through a transcriptional mechanism, that would have to be associated with a change in chromatin associated with an increase in gene transcription. So it might be an increase in histone acetylation, an increase in a certain type of histone methylation, as just some examples, or a reduction in some repressive chromatin mechanism. And we would identify these measures genome-wide by use of another approach called chip sequencing, chromatin immunoprecipitation, followed by sequencing, or related methods that are now used. And we would overlay still additional data sets on top of this by focusing on types of transcription factors that we think mediate that opening and closing of DNA. And here are just some of the names of transcription factors that we've studied in the context of chronic stress. So this is an enormous undertaking. Just the data represented by this slide, a single brain region, a single time point, looking at these measures would generate about 20 terabytes of sequencing data. And this is an enormous bioinformatic challenge. Honestly, the field is still figuring out how to optimally use these large data sets. I'll give you some examples today of a progress report. So the first study that I'll mention was done by Rose Baggett in the lab. She's now back at McGill in Montreal, where she subjected mice to chronic social defeat stress, looked at them 2 days after the last stress, separated resilient and susceptible animals, performed RNA sequencing on 4 brain regions shown here. We have other brain areas that are not depicted: nucleus accumbens, medial prefrontal cortex, basolateral amygdala, ventral hippocampus. Yellow shows genes that are upregulated, blue genes that are downregulated. For those of you who are not familiar looking at these heat maps, every single vertical line represents a single RNA, or the product of a given gene. The Venn diagram at the right shows the same data, just comparing the total number of genes that are affected in the resilient condition versus those in the susceptible condition. And just to give you an idea of how we use a high altitude analysis of these large data sets, for example, when we started embarking on this work, we hypothesized what would make an animal resilient? Well, it fails to show the bad effects of stress. And that does happen to a certain extent, as depicted by the Venn diagrams, but the predominant pattern is different from that. The predominant pattern is actually many more genes are regulated in resilience in most brain areas compared to susceptibility, leading to an additional hypothesis that perhaps resilience is the more plastic adaptive state, and that susceptibility results from animals that are inherently less adaptive, less plastic. Now, because of the concern about the accuracy or validity of an animal model, we have paid increasing attention to validate studies in animals by direct study of postmortem human brain tissue. And this shows the results of a study led by Benoit Labonté in the lab. He's now back in Laval University in Quebec. And what Benoit did was to analyze 100 human brains obtained from two brain banks, one Gustavo Turecki at McGill, the other Carol Taminga in Dallas, half depressed, half not depressed, half men, half women. So for a heterogeneous syndrome, a small data set, but the biggest data set available for human depression. We did RNA sequencing on these 100 brains for 6 brain regions, several regions of prefrontal cortex, nucleus accumbens, and ventral hippocampus. I'm showing you genes that are up or down regulated in one brain area, medial prefrontal cortex, of depressed men, and asked how those genes vary in other brain areas, same for the females. And again, a high-level altitude analysis shows an interesting difference. Notice how the genes that are induced or repressed in males are largely different in these other brain areas, where in the depressed female, many of the same genes are affected across the brain. I'll come back to that in just a moment. The availability of these large data sets also makes it possible to validate an animal model, not only by comparing behaviors, as I mentioned earlier, but by comparing transcriptional profiling, asking the question, to what extent do different chronic stress models in a mouse recapitulate the gene expression abnormalities that we find in depressed humans? And that's a study that is now underway by Benoit, just showing you examples of prefrontal cortex, nucleus accumbens. The diagram is meant to be heuristic, not quantitative, basically stating to what extent 3 chronic stress models, I've already mentioned social defeat, another is social isolation, a third is chronic variable stress, which I'll come back to in a few minutes, in mice, asking to what extent do each of these recapitulate gene expression abnormalities by sex, by brain area, with human depression. And what we found is that no model is appreciably better than other models. But interestingly, each model recapitulates about 25% of the gene expression abnormalities seen in human depression, but largely a different 25% in each model. Maybe that would be expected for a syndrome as heterogeneous as depression, but we're very interested in the insight that these kinds of comparisons can provide. Okay, so with that as a way of introduction to the broad topic that I'd like to discuss today, I'll talk about 2 specific projects that are in progress. First, talking about sex differences in depression, and the other, if there's time, looking at mechanisms by which stress early in life renders an individual more susceptible to depression for a lifetime. First, sex differences. We know that depression is more than twice as common in women than in men. Sex differences have been reported in clinical features of the syndrome and treatment responses, but by and large is poorly understood at a mechanistic level. So we looked first at our RNA-seq data sets in humans. So this is going back to the RNA-seq data that I already showed you from our human data set. now directly comparing genes that are affected in depressed men versus normal men with those genes that are affected in depressed women versus normal women. And what we found was an astoundingly low level of overlap, less than 5 or 10% overlap in these gene expression abnormalities between males and females. This is not seen in other psychiatric syndromes, for example. This was a surprise. We were very concerned that this might reflect a bioinformatic artifact of comparing large data sets. So we went back to an animal model to directly compare males and females. And I should add that this lack of overlap in human depression was seen across all the 6 brain areas that we studied. So we turned to a different chronic stress model, chronic variable stress. Some people refer to this as chronic mild stress or chronic unpredictable stress, because work done by Georgia Hodis, who's now at Virginia Tech, then in Scott Russo's lab at Mount Sinai, working with Benoit, had found that this model recapitulates the greater susceptibility of females to certain types of stress with respect to certain types of endpoints. That's depicted in this slide, looking at animals subjected to a different stress every day, either for 6 days or 21 days, showing you 4 behavioral outcomes. We have others. I'm not going to explain what each of these mean. I'd be happy to during the question period if you're interested. Showing that it takes a full 21 days for the male animals to develop behavioral abnormalities, whereas in this assay, and with these behavioral endpoints, the female mice develop maximal behavioral abnormalities after only 6 days of stress. So what we did was to carry out RNA sequencing after 21 days of stress when the males and females displayed equivalent behavioral abnormalities. And what we found was similar to what we had seen in human, a relatively small overlap between chronic stress-induced gene expression abnormalities in the males versus the females. The overlap was bigger than in humans, maybe 10 or 20%, but I want to emphasize these are genetically inbred animals subjected to identical stresses, with the males and females exhibiting equivalent behavioral abnormalities, and yet gene expression changes overlap by less than 20% across brain regions. So we believe that the findings from our human data sets are real, and they suggest that depression is a fundamentally different syndrome in men versus women, and it argues for a sex-specific approach to understanding that syndrome, something that has not really been undertaken to date. Now, all the data that I've shown you thus far has been analyzed in a relatively simple way, just asking a simple question: is gene X expressed at a different level under condition A versus condition B? But the size of our data sets allow us to employ more complicated bioinformatic approaches, such as looking at gene co-expression networks. For this, I want to thank Bin Zhang, who's been a great colleague at Mount Sinai, and his mentor, Eric Schad, who introduced us to these more complicated bioinformatic approaches. So weighted gene co-expression network analysis is an example of these approaches. It simply takes every transcript in a data set, perhaps 30,000 transcripts, and ask, what is the relationship of each transcript to all of the others, across brain regions, across time points, across species, or sex, and collecting the genes into individual modules where each module contains maybe 100 up to 1,000 genes that are correlated within our data sets. The construction of these modules is solely based on our data sets. They have nothing to do with the known biology of these genes. I'm depicting these modules in a so-called Circos plot, where each slice of the pie is a gene module. It goes from the most significant to the least significant as you move counterclockwise around the pie. We can, the concentric circles then ask, to what extent do the genes in a module also show differential expression after stress or in human depression, across different brain regions in the female brain or across different brain regions in the male brain. So the most significantly enriched module contains genes that are enriched for synaptic transmission. It's interesting. But it looks like it's equally implicating genes affected in the female and male brain. In striking contrast, the next most significant module enriched for genes that are involved in MAP-Kinase signaling, I'll define what I mean by that in a moment, are dramatically enriched for genes that are differentially expressed in the female depressed brain, much less so in the male depressed brain. So we focused on that module. This is a depiction of the hub genes in that module. Hub genes are simply genes that are more correlated with other genes within a different module. Again, based solely on the correlative data within our data sets, not based on anything known about the biology of the gene. The most highly ranked hub gene in this module is a gene called Dusp6. It encodes a MAP-Kinase phosphatase. I'll define that in just a moment. And this was by far the most highly ranked, differentially expressed gene. It was differentially expressed only in the female depressed brain across multiple brain areas, an effect also seen in mouse prefrontal cortex. Now, Dusp6, as I mentioned, encodes a MAP-Kinase phosphatase. A MAP-Kinase is a major class of protein kinase present across tissues, including in brain. A MAP-Kinase phosphatase removes phosphate groups from that MAP-Kinase and inactivates the MAP-Kinase. So the first step in validating these data sets is to look at a completely independent cohort of human and mouse tissue to validate the findings, see if it's replicable. The findings are replicable in ventromedial prefrontal cortex. Levels of Dusp6 expression are reduced in female depressed patients, not in males. Similar effect seen after 21 days of chronic variable stress. We wanted to see if this molecular change has some expected biochemical consequences. So we looked at an enzyme called ERK. ERK is the most important type of MAP-Kinase in the brain. We looked at levels of phospho-ERK. Again, if an ERK phosphatase is decreased, one would expect increased levels of phospho-ERK in the tissue, and that's what we see. That in depressed humans and in chronically stressed mice, there's an increase in phospho-ERK in ventromedial prefrontal cortex in females, no effect in males across species. We can also use immunohistochemistry to identify the cell type within the mouse and human ventromedial prefrontal cortex that shows this change in phospho-ERK, and it dominated in pyramidal neurons within this brain region. Now, everything that I've mentioned thus far is correlative. It's just based on a deduction, a bioinformatic deduction on our data sets that Dusp6 is part of this module. So we feel that it's essential to validate these bioinformatic predictions empirically. So to do that, what Benoit did was to construct a viral vector that makes what's called a microRNA, a small sequence of RNA that targets Dusp6, injected that virus in the ventromedial prefrontal cortex of mice, and asked to what extent does manipulation of Dusp6 alone, right, replicating a stress-observed decrease in expression levels of that gene, replicate the other gene expression abnormalities seen in the tissue. So what we did was to inject the virus into that brain area and then did RNA sequencing on the infected tissue. What I'm showing you here is called a rank-rank hypergeometric overlay analysis. It provides a very nice way of comparing two large data sets in a threshold-free manner. So what we're asking is, to what extent does knocking down Dusp6 in the female ventromedial prefrontal cortex replicate changes in gene expression seen in that brain region of female mice exposed to chronic variable stress? And you can see a robust overlap. And if one drills into the genes that comprise that overlap, there's dramatic enrichment of this module, replicating the downregulation of Dusp6 and up and down regulation of other genes in that module, providing direct empirical evidence that Dusp6 really does function as a hub gene in practice, not just in theory. We then use this same viral approach to ask whether this is a behaviorally relevant effect. Is this related at all to depression? So what Benoit did was to inject viral vectors expressing this microRNA that knocks down Dusp6, or a control vector expressing green fluorescent protein alone, subjected animals to what we call subthreshold chronic variable stress. This is a level of stress that has no effect in normal males or female mice. But in the context of a knockdown of Dusp6, this low level of stress can produce significant behavioral impairments in females only. No effect seen in males, even though we can show that the knockdown is equivalent in the two sexes. We can show the converse type of information now using a viral vector to overexpress Dusp6. This would essentially rescue that downregulation caused by chronic stress of this gene, subjecting animals to the full course of chronic variable stress that induces behavioral abnormalities in both sexes, showing that rescuing Dusp6 rescues the female animals behaviorally with no rescue effect seen in the males, again, despite the fact that the gene is overexpressed to the same extent in the two sexes. In order to gain some initial insight into the mechanism underlying this effect, we turn to a friend and colleague, Yan Dong, at the University of Pittsburgh, who recorded from pyramidal neurons in ventral medial prefrontal cortex of animals that had induced lower levels of Dusp6 using our viral approach. The viral approach has no effect on the excitability of pyramidal neurons in this tissue. However, knocking down Dusp6 increases this one measure of excitability in the female neurons, but not in the male neurons. So this is where this work sits at the moment. We're very interested in understanding why it is that the same biochemical intervention in the same nerve cell type, in the same brain area of a male and female brain, can produce such a dramatic effect in one sex but not in another. Underlying mechanisms remain unknown. One possibility is that these dramatic sex differences seen in human depression and chronic stress in mice reflect hormonal differences. They could reflect chromosomal differences. They could even reflect in differences in life experience, particularly in humans, but perhaps in mice as well. So that's something that we're very interested in studying. Now, the question is, if human depression is so different, as I'm telling you that it is, in males and females, why is it that most antidepressant drugs work about the same in the two sexes? We would give a few answers. First of all, even though the overlap in gene expression abnormalities is only 5 or 10%, that's still hundreds of genes that are regulated the same. Many of the genes that my lab has studied previously are regulated equivalently in both sexes. In other cases, even where different genes are affected in males and females, they influence the same biochemical pathway. So there is overlap functionally. Nevertheless, we believe that there are fundamental differences, like in MAP-Kinase signaling, as I illustrated for you today, which is just one of now many examples of fundamental differences in biochemical pathways affected in human and human depression and mouse chronic stress in males versus females, and it does provide a path forward for a sex-specific analysis of antidepressant drug development. Okay, with that, I'm going to pivot in the remaining 10 or so minutes to tell you about the other point that I raised earlier in understanding how it is that early life experience can program an animal's behavior for a lifetime. So we know that the strongest risk factor for human depression is exposure to adverse life events in early childhood, but the mechanisms remain poorly understood. We were interested, and this is all work done by Kate Pena in the lab, to develop a model where early life stress would not produce lifelong changes in behavior per se, but a lifelong susceptibility to subsequent stress. So a two-hit model. Why? Because most early life stress models in rodents have found robust effects for a lifetime in the rodent's life, but that's really not what we see in most humans. Most humans exposed to early life childhood do just fine. They're just more vulnerable to stress later in life. And so what Kate did was to embark on looking for a mouse model that would recapitulate that phenomenon. And what she did was to separate animals a few hours a day from their mothers, either in the first half or the second half of that pre-weaning period, while also reducing the amount of bedding material available to the mothers. And what she found is that exposure to this form of early life stress really in the second half of this pre-weaning period produced the effect that we wanted. No effect on behavior in the absence of subsequent adult stress, but an increased stress susceptibility seen in adulthood. This composite measure shown in the middle panel is probably the easiest to recognize. No effect of early life stress when the animals are not exposed to a second hit. Following chronic social defeat stress, evidence of stress susceptibility in normally reared animals and those exposed to early life stress in the early part of the pre-weaning period, but a dramatic increase in susceptibility with that later term of stress. So we decided to better understand this phenomenon. And the same thing occurs in males and females. So what we did, which is our standard approach now in the lab, is to do RNA sequencing on several brain areas. I'm showing you 3 brain areas here. And in this case, I'm comparing 2 conditions to make a point. I'm comparing the effect of this early life stress to changes in gene expression that last a lifetime. So these animals were analyzed when they were adults but had not been exposed to a second hit, versus animals that were raised normally, no early life stress, but exposed to a full course of chronic social defeat stress or another form of adult stress. And what we found in each of these brain areas is some similarity between these conditions, suggesting that one thing that early life stress is doing is to actually priming these brain areas transcriptionally to look like gene expression patterns that occur after a full course of adult stress. This effect was most dramatic statistically in the male ventral tegmental area, and we can show that similarity using the RRHO analysis that I mentioned earlier, comparing the effect of early life stress by itself, social defeat by itself, seeing a highly significant overlap between these 2 data sets. And if we look into the genes that comprise this overlap, a gene was deduced as a top upstream regulator. In other words, of all the genes that share this regulation in common, OTX2 is predicted to be upstream of regulating those genes. OTX2 is a transcription factor that has been implicated in brain development, including VTA dopamine neuron development, but never implicated in stress responses. So Kate decided to study that further. She looked in our RNA-seq data sets and in fact found, as an example, and I'm just showing you some examples here, of 6 genes that are predicted as OTX2 targets after early life stress, all of which show a downregulation in adulthood, even though OTX2 levels by themselves are not affected. So that was surprising. OTX2 is a predicted regulator. The downstream genes are affected, but OTX2 itself is normal. So what Kate did was to look earlier in development and documented a decrease in OTX2 levels immediately after this period of early life stress, shown at the RNA level, also at the protein level, with levels of OTX2 returning to normal in adulthood, even though expression of these target genes remains impaired for a lifetime. We can also use this analysis to confirm that virtually all OTX2 expressed in the ventral tegmental area is present in dopaminergic or Th-positive nerve cells. We wanted to test the behavioral relevance of regulation of OTX2, so Kate designed, very analogously to what I described for the other project, a herpes virus that expresses OTX2. In this case, we used a herpes simplex virus, which is perfect for this experiment because herpes simplex viruses express their transgenes for just a few days. So it's a way to overexpress or knock down a protein for a few days without affecting that gene for the rest of the animal's lifetime. So in this experiment, Kate exposed animals to early life stress. At the end of that period, injected this herpes virus, overexpressing OTX2, rescuing that transient decrease in OTX2 without affecting OTX2 for the lifetime, and found, in fact, that that produced a behavioral rescue as well. Exposure to early life stress, overexpressing a control virus, showed an increase in susceptibility of these animals that I showed you before. Rescuing OTX2 just during that early developmental period rescued the animals behaviorally. And we found in the same animals evidence of downregulation of those same 6 example predicted downstream target genes of OTX2, with expression of OTX2 early in life rescuing adult expression levels of these genes. Kate did the converse experiment, now taking a herpes virus expressing a microRNA that downregulates OTX2. In this experiment, the animals were not exposed to early life stress. They were just given this viral treatment that knocks down OTX2 very transiently, just for a few days in development. OTX2 is then normal for the rest of the animal's life. What we were able to show is simply knocking down OTX2 for a few days during development is enough to induce stress susceptibility. And in fact, it was also enough to produce a significant lifelong downregulation of a few of these target genes, not all, but several of these target genes. So a transient manipulation of OTX2 early in life produces a permanent impairment in the expression potential of these genes. We've more recently looked at this genome-wide by doing RNA sequencing on the tissue. And what I'm showing you by heat map and by Venn diagram is a very large degree to which this transient juvenile knockdown of OTX2 in the ventral tegmental area recapitulates a large fraction of genes that are affected just by early life stress. So we think that that OTX2 is a dominant mediator of this effect. Now, looking at several of the genes that are affected are many ion channels and transporters, putting us in a direction of looking at VTA neuron excitability. I'm not going to talk about that further today, but it's an area of interest. So as my last data slide, we are very interested in why it is, how, by what mechanism does this transient reduction in the transcription factor for just a few days produce a permanent impairment in regulation of downstream targets of that gene. So we conducted an early experiment called quantitative CHIP, or Q-CHIP, quantitative immunoprecipitation, looking at putative target genes from our RNA-seq datasets, the same genes that I've mentioned earlier. What we're showing here is the degree to which OTX2 is binding to specific active sites within these genes early in development and later in development under standard or early life stress conditions. What we found, what Kate found, is that there is appreciable levels of binding of OTX2 to each of these target genes that validates the bioinformatic prediction. And notice how exposure to early life stress dramatically depletes OTX2 binding to these genes at this early time point. That makes sense. OTX2 levels are down. Its binding to target genes is down as well. Surprisingly, however, in adults, this binding is fully restored. In fact, there's even evidence that there might be an overshoot, increased binding of OTX2, although that's not statistically significant. So even though OTX2 binding recovers, these genes remain impaired. Why? What is the so-called chromatin scar at those genes? Well, we have some insight into what those chromatin scars might be using a proteomic approach, taking VTA tissue from animals subjected to early life stress or standard conditions, collaborating with Ben Garcia and colleagues at the University of Pennsylvania. We were able to find a few histone modifications that are altered in this brain region. I'm going to just give you the code. This means mono or trimethylation of lysine 4 of histone H3 or dimethylation of lysine 79. Doesn't matter what this means. 3 different types of chromatin modifications out of many hundreds that are detected in this proteomic analysis. We're now doing CHIP sequencing for these particular marks. Here's an example of a CHIP sequence for this one mark, showing, as predicted by the proteomics, a genome-wide enhancement of this mark across genes. TSS stands for transcription start sites, transcription end sites. So you can see this genome-wide enrichment. This is just an example of enrichment at one particular gene. We're now overlaying these ChIP-seq data sets, for example, taking all the genes that show an increase in this histone mark as a result of early life stress, asking whether the same genes are affected in terms of their RNA expression after early life stress or after early life stress plus adult stress. And we're seeing interesting statistical correlations between these various measures, something that we're now very interested in studying in greater detail. So we're particularly interested in this model because we think it represents a nice heuristic way of looking at how it is that stress early in life changes specific types of nerve cells for a lifetime, really drilling in at a very reductionistic level, looking at gene expression and transcriptional chromatin structure at individual genes. We think that OTX2 is particularly important in the male VTA. It is not implicated in the female VTA or in other brain areas. Other mediators are responsible for a similar pattern in those brain regions. And we're very interested in identifying these chromatin scars. How is it that a reduction in a transcription factor early in development targets that gene, marks that gene for a lifetime to show impaired expression? So with that, let me conclude by saying that we think that studies at the transcriptional and chromatin level, both in mouse models and in humans, will teach us a lot about the neurobiology of stress responses and depression. I focus today on mechanisms of stress susceptibility and resilience, focusing on sex differences and lifelong control of stress susceptibility. We're also interested in looking at mechanisms of antidepressant action. The hope is that this work will lead us to improved diagnostic and better treatments for depression, although I have to say I've had a lab for 30 years. We all thought at the time that this was going to be easier than it's proven to be. My field has largely failed to advance treatments, and so the young people in the audience will have to do much better than my generation has done in taking this increased knowledge of fundamental biology into the clinic. So I'll stop there, and if there's time, I'd be happy to answer any questions. Thank you.
So there would be an argument, one might suggest, for whether transient treatment with epigenetic drugs such as histone-deacetylase inhibitors, DNA-methyltransferase inhibitors might block that later susceptibility.
Right.
Have you had a chance to look at that?
Yes, we and others have published a few studies on histone-deacetylase inhibitors, either direct administration into the brain or, in some cases, peripheral administration. And they do produce an antidepressant, pro-resilience type effect across several models. The challenge in advancing that approach to humans, however, is that, and there are several histone deacetylase inhibitors that are in trials for human cancers, is that they're toxic molecules. Histone deacetylases are expressed in every tissue in the body. They produce robust effects, not surprisingly. Chronic inhibition of HDACs, histone deacetylases, produces many off-target effects. So the trick would be to identify inhibitors that are targeting brain-enriched proteins. That's work underway, but really there's been no chemical progress yet in doing that.
Good.
So fascinating. So I guess if you take the females in your first study and breed them and look at offspring, any indication of transgenerational?
Right. So we.
Some other systems you well know.
Yes, absolutely, and certainly for nutrition, that's been one of the fields where transgenerational transmission has been demonstrated most robustly. We've looked at transgenerational transmission a little bit, but only paternal transmission. We have not looked at maternal transmission, just because it's one generation harder to prove. And there is now evidence across several labs that stressing an adult male mouse will lead that mouse to breed male and female pups that show greater stress susceptibility. The key question in our minds is mechanism. We have evidence that part of the mechanisms may be behavioral. Turns out that there's a large literature in animal psychology that mothers give their children different resources based on the robustness of the male with which they had sex. For example, certain species of female birds give the size of the yolk is related to the robustness of the bird feathers and bird song of the fathers. Crazy stuff. So one of the ideas is that perhaps the mothers are giving different resources to the pups, and that explains the transmission. But the best work has been done by Tracy Bale, used to be at Penn, now at Maryland, who has provided evidence that there is regulation of specific microRNAs on sperm cells of stressed male mice, and that if she recapitulates altered levels of expression of those particular microRNAs within a zygote, she is seeing increased stress susceptibility in the adult animals. Now, how you go from those microRNAs to a brain-specific phenomenon is a big question, but at least it provides a pathway forward.
Here and then go ahead. I just have a question. How do you choose the specific brains? So there are a lot of brain areas about them, like depression, you got diagnostic, ILC,
Right.
a bunch of them, right? So, but you just focus on a few of them. What
Right.
makes you do you ignore the other ones, or do you just focus on these few brains?
The approach that we've taken, to be honest, is to analyze as many brain regions for which we have money. And if we had more grant money, we would do additional brain regions. That RNA-seq study that I mentioned is the largest one done to date in human depression, is a 600 sample RNA sequencing experiment. So do the calculation of $1,000 per lane of sequencing, right? So you get a big number. So we focus on regions that we think are important, but there are many additional regions that would also be worth studying. I'll make it even more complicated for you. All the sequencing that I've talked about today has been done on crude tissue extracts, whole extracts of these brain areas, whereas we know that each brain area contains many different types of nerve cells as well as non-nerve cells. Each cell type has a distinct pattern of gene expression. And so we're now repeating many of these studies looking at specific types of cells. One can reveal still additional changes. So one really needs to do these experiments at the level of specific cell types across the entire brain. We really don't have the feasibility to do that yet.
Next here, and then we'll go Joe and Zenya.
Yeah, that early slide you showed of the brain circuitry of depression indicates where you put your money. Why does that look so similar? It looked like it could have been the introduction to a talk on addiction.
In terms of the brain areas?
Yeah,
Oh, yeah, yeah.
what's your take on that similarity?
Well, so, we refer to them as limbic brain areas. That actually just is some old a term in German by German neuroanatomists. But it reflects the entire forebrain and the fact that the forebrain brain areas are connected among each other in this highly interacting broad circuitry that are important in emotional behavior. So they've been implicated not only in addiction and depression, but also schizophrenia, Alzheimer's disease, and the list goes on and on. I mean, this is the part of the brain that is most dramatically developed in humans compared to other species, where we think a lot of the neuropathology exists.
Joe?
Hey, Joe.
I have two questions. One, in the very early part of your talk, you talked about the observation that there was resilient genes they seem to turn on. Is it possible not to worry about the mechanism and just look for something that turns on those resilient genes?
Absolutely. Yeah. So let me answer that because I'll forget it when you go to the second one.
You have to go after the mechanism, but
Yeah. So in the psychiatric and neuropsychological literature, there's a great deal of interest in training humans to be more resilient, and one need not know the mechanisms in order to do that. And there are now many programs that people follow to build resilience. Probably the best established is one used by our armed forces to train special forces troops, Navy SEALs, Army Rangers. These people are extremely resilient, right? The Navy SEALs get waterboarded, right, as part of their routine training. So one way to build resilience is to subject people to a stress that's high, but not so high that they can't take it. Of course, that varies from person to person, and do that chronically. And there are other approaches as well. There's a lot of interest in that.
the second question, you mentioned at the very end of your talk that your field has failed in certain ways, but I believe that drug companies trying to find an appetite suppressor has failed and probably have spent more money, $50 billion in research over the last 50 years. and they have yet to find
Right.
a safe appetite suppressor.
You know, I appreciate you saying that, because sometimes I'm too hard on my own field, and other fields have seen a similar difficulty in translation. But when I think across my lifetime, there are certain categories of illness, cardiovascular disease, immunologic disease, probably more than anything else, where our treatments today are amazing compared to what I learned in medical school 30, 40 years ago. And so there has been incredible progress in some fields. I agree with you, not so much in obesity, but in some fields. And psychiatry, obesity, certain other areas have not shown the same kind of advances. So we need to figure out why. Maybe doing the same thing over and over again is not good, and we need to try different things, is what I would say.
All right, we have time for one more. Tanya?
Thank you very much for that interesting talk. I'm wondering if I can go to an area that we're interested in the role of the microbiome.
Yeah, sure.
I am
Yeah.
wondering, now that we're starting to see that new effects are related to the microbiome, have you done any research with mice that are sterile and done some?
Yes. So we're very interested in it. This is not work that my own lab is doing. Vanna Zachariou and Patrizia Casaccia and Drew Kirley at Mount Sinai are all investigators focusing on this. Not surprising to you guys, if we treat mice with oral antibiotics that are not absorbed to deplete most gut bacteria, we see dramatic effects on complex behavior. So depending on how it's done, the animal's susceptibility is dramatically altered to stress as well as to drugs of abuse. And the key question for me is the mechanism for how that is happening. And that's something that we don't yet have, I think, good insight into, but it's something that these groups at Mount Sinai and other places are very, very interested in doing. It is phenomenal. Now, it's interesting from a historical perspective in terms of the gut microbiome. When I was in training, it was something we never thought would influence complex behavior with a size signal that we know it does. And now today, this is a new robust area of biology that I think is very, very exciting.
In addition to a classic nutritional sciences water bottle and a Seb's bag, we have this plaque in recognition for the Hans Fischer Lectureship entitled Transcriptional and Epigenetic Mechanisms of Depression by Eric J. Nestler, MD, PhD, April 18, 2018.
Thank you
Thank
so
you
much.
very
I really appreciate it. Thank you. Really appreciate it.